Hello, everyone. It is of great pleasure to be invited here to present at the Lab Roots virtual event of clinical diagnostics and research. My name is Jing Cao. I'm an Associate Professor of Pathology at the University of Texas Southwestern Medical Center in Dallas. And my topic today is circulating biomarkers of traumatic brain injury. Here's my related disclosure for this talk. I do receive research funding from the National Institute of Health on traumatic brain injury diagnostic markers. Here are the learning objectives of this presentation. We will try to understand the global impact of TBI and its long-term consequences. We will also review the current diagnostic approaches and explore their limitations in accurately characterizing and timely identifying TBI. Then we will focus on the field of non-invasive blood-based markers of TBI, including proteins, nucleic acids, and a vesicle called extracellular vesicle, and its associated markers. The acute encounter of traumatic brain injury affects approximately 10 million people annually worldwide and is a leading cause of disability. So it is defined as structural damage or disruption of normal brain function, and it may result from head impact, shaking, or penetration. And then depending on the duration of loss of consciousness, post-traumatic amnesia, and then altered mental status and neuroimaging findings, we can have different severity from mild to severe of traumatic brain injury. So beyond the acute phase of TBI, this condition is actually now considered a chronic process. It does have long-term effects, could be depression, aggression, psychosis, traumatic stress disorder, etc. And some of the neurodegenerative disorders are also associated with previous TBI event. Those that we are quite familiar with are Alzheimer's, Parkinson's. So recurrent TBI, as we observe in athletics and in field injuries, these can lead to persistent or worsening neurobehavior symptoms. So I think a well-known one is the PTSD in our soldiers, and that's possibly related to their traumatic brain injury. So there are a variety of severe long-term effects of TBI, and we're listing the major ones here, including memory loss, headache, epileptic seizure, dizziness, visual changes, fatigue, and etc. For brain injury, here I'm using the analogy of two other major organs, our heart and liver. So people or professionals in the laboratory medicine field will know for cardiac or heart disease, and as well as for liver disease, we all have well-studied extensive list of biomarkers. For example, for heart, when people have an acute myocardial infarction, we could have troponin for diagnosis. And for people with heart failure, we could have BNP or NT-proBNP as the biomarker. And the same for liver disease. We have a list of liver enzymes. We can also look at bilirubin for cholestatic disorder. And then we can look at the function of liver by examining albumin or clotting factors. However, for brain, which is a organ as vital as liver and heart, if not more important, actually there is really scores of biomarkers available for us to provide an objective evaluation of its injury and function. For traumatic brain injury, currently in clinical practice, we do have multiple modalities trying to diagnose it, including imaging and clinical assessment. And also we can use electrophysiological methods like EEG to help us understand the nature of the injury. So clinically, the injury usually is categorized based on a score system, and a widely used one is the Glasgow-Coma scale. The severe ones are 3 to 8, and the mild ones are with higher scores from 13 to 15. However, the clinical assessment can actually differ from actual structural pathology. If we look at the figure on the left, we could see the top two brain images are from, one is from mild, the other is from severe TBI. If we neglect the cavity on the left side, you will find these two actually brain structural pathology are quite similar, maybe some edema, but not much really significant difference. And the cavity that we found on the left actually is from a previous loss of brain tissue. So from even with the CT high resolution imaging methods, we are not really able to tell the difference between mild and severe. So when we're trying to identify valuable or instrumental biomarkers for TBI, we first need to understand this process. It is not a straightforward, simple injury. It does involve multiple cell systems, and it has multiple pathways actually will affect our study target for biomarkers. So it can be roughly divided into four major categories, one related to neuronal cell death, the other related to the blood-brain barrier breakdown and edema. Also, we could have inflammation going on with it, so releasing a variety of inflammatory biomarkers and mediators. And there are efforts going on in the research field trying to understand TBI and also provide better management because of the threat that it poses. So in both the United States and Canada, here in North America, as well as in Europe, there are actually a consortium of study centers that try to leverage the resources of patient and medical resources. So we do have multiple studies going on, and there are actually some of the results studies have come to fruition out of these networks. The most studied biomarkers of TBI are mainly proteins. And here are a list of them, and we will go into each of them just to introduce its basic features. So including the GFAP and NFL, the ubiquitin C-terminal hydrolase, the S100B, and tau proteins. Other biomarkers studied include markers of inflammatory responses and vascular injury, and also circulating microRNAs. Small non-coding RNA can also play a major role in the regulation of gene expression. Let's look at a list of proteins first. So the glial fibrillary acidic protein is a component of the intermediate filaments in astrocytes. So the GFAP monomers, they homopolymerize to form the intermediate cytoskeleton of astrocytes. And during astrogliosis, they are upregulated. And then after traumatic brain injury, they are also released from astrocytes. So we will find its elevation in both CSF and blood. The next marker is the NF light chain. So the neurofilaments, they overall provide a framework within the neuronal axon. And this protein comes in three forms: heavy chain, medium chain, and light chain. So those neurofilaments in the range of 10 nanometer diameter range, they are formed, the backbones are formed by the polymerization of the neurofilament light chains. And they have a pretty long half-life of 3 weeks. And again, in both CSF and blood, the NFL light chain was found to be increased after traumatic brain injury. The ubiquitin C-terminal hydrolase L1 is an enzyme that's highly expressed in neurons. So as the name implies, it is involved in ubiquitination and deubiquitination of proteins. And this protein is also found to really transiently increase after brain injury. The next one is S100B. It is a calcium binding protein with pretty low molecular weight. It's expressed in glial cells and neuroleptic cells. It has a very rapid increase after injury, but it's also very rapidly eliminated from the blood. So you can see its half-life is quite short, up to 2 hours. But there's a really huge magnitude of increase, can be up to 100 times higher after the brain injury in the CSF than in the serum. And tau protein is already a very promising biomarker in neurodegenerative disease. It's also dysregulated in traumatic brain injury. So it is a protein involved in stabilizing the microtubules in the neuronal axons. It has, again, a number of isomers and molecular weight ranging from 33 to 46. So after the brain injury, we could have multiple forms, actually, including cleaved tau, phosphorylated tau, and total tau. So just to keep in mind that healthy neurons also release tau into the interstitial fluid, and the turnover is slow with a very prolonged half-life. So it's not a single target, but could be an array of different tau proteins. Here is a theoretical kinetic curve of these biomarkers, and the data are a combination of mouse cells and human studies. So there are markers that come and go very rapidly, and the S100B we mentioned is among one of them. And on the other side, we could have very long tails, that those are markers with very long half-life and just stayed in the system without being cleared for a long time. So many of us in the diagnostics field, we know that currently there are already FDA-approved automated assays for traumatic brain injury. So the two markers selected are the GFAP and the UCHL1. So the utility of these two biomarkers is actually interesting, focusing on the mild TBI, and it's being used as the claim is that you can use it within 12 hours of injury, and then to determine the need for CT. In other words, is that if you do have positive finding on these two markers, then it's likely you will also have a positive finding on the CT, so it's necessary to go ahead and send the patient for a CT scan. But if you do have a negative result, then it's not likely that you will have a positive CT finding. However, as we learned from previous slides, even the CT finding is not a gold references, because there are severe TBI with very minimum actually structural pathology change. And also these markers are only available in serum, not in whole blood yet. So it requires an extra centrifugation and separation step. Having covered protein biomarkers for TBI, the next several slides I'm going to move on to talk about a vesicle that could be a promising target for TBI marker. That is the exosome, extracellular vesicle. So they may contain cargoes that play a direct role in brain injury pathology. So the scale or size of exosomes are between 100 and 1,000 nanometer range in diameter, and they are actually double layer vesicles formed from the bubbling of organelles and cell membranes. And so there could be the budding from plasma membrane or fusion of multivascular bodies, and then they will be released into the extracellular space. So these vesicles, they have surface protein derived from the parent cells. Also, they serve as a vehicle that can carry proteins, mRNAs, microRNAs within the lumen, and it reflects the state of their cell of origin and can be transferred to the recipient cells. They cross the blood-brain barrier, making it a very actually useful biomarker for monitoring the progression of traumatic brain injury without the really need to have an invasive technique to look into the CSF. So in this slide, I'm showing that there are multiple actually traditional, conventional ways to isolate these IVs using either affinity or separation techniques. But they all require pretty large volume of fluid, and also it's not really practical in clinical settings because it's manual and tedious. There have been efforts to bring the EV testing to reduce its volume requirement and also maybe bring it to a near patient site. There are multiple attempts in the scientific field, and I particularly want to introduce this one from Dr. David Isodor's group at the University of Pennsylvania. So they have a study, actually, they used patient versus control samples, and they kind of developed this technique called track-etched magnetic nanopore technology to be able to isolate out the exosomes. and it's based on a antibody binding to enrich actually CSF originated exosomes. In their previous study, this group has looked at promising protein biomarkers, and they were able to actually draw a line between control and patients to say that yes, they do find a significant higher content of these mostly inflammatory markers in the case versus control. Then in the current study, they again used this tempo technique and used the GLUR2 to enrich for brain-derived EVs. So it is a receptor subunit that is found preferentially in brain-derived EVs, so that they can enrich this subset of EV and then to look at its signature pattern. In this study that they particularly looked at microRNA markers, they had mouse models as well as clinical samples. So basically, the methodology was developed from the mouse model, and then they tried to transfer to the human. However, in human actually clinical sample, they had to actually use another microRNA set. But they do try to characterize different injury types and different severity, so that when we're trying to utilize these markers, we can not only just to say yes or no, but to say how the injury is being characterized. So that is, I think, a breakthrough of this study. So not only to be a absent or present diagnostic marker, but also to be injury type and pathological process specific biomarkers. So in the study group, they had adult patients with different Glasgow Coma scales, and then they collected plasma sample from these patients. A graphic summary of their study, they do find a distinct expression pattern of a panel of microRNA, and then they also used the sensitivity and specificity based on true negative and true positive, false negative, and false positive results to construct this ROC curve. You can see it's much better than a neutral curve, ROC of 0.5, but the ROC here is quite close to 1. What's exciting about the study is that they can have a multiplexed microRNA biomarker to specify specific injury state. And this technique does use substantially lower sample volume, although it's still 10 ml, quite a lot, and it's a short time period to run the isolation. However, the antibody they use to enrich for EVs, it's only limited to certain cell types, and the condition of this EV microRNA is still quite stringent, the storage condition, because they have to process it on site immediately. And also, the microRNA panel is hard to translate across organisms, because like from mouse to human, we could have a different panel. So we have to really study human samples to find out the unique signature of TBI. And there are other emerging techniques. One of them that I'm working on is called liposome-mediated isolation of extracellular vesicle. And this started actually from a collaboration with Dr. Tony Hu's group at Tulane University. We initially used this technique to study SARS-CoV-2 RNA-positive extracellular vesicles. So we synthesized these, again, nanoscale liposome that it can fuse with EVs, and this liposome contain actually a machinery of molecular tests reagents, so that the reaction will occur in these fused vesicles, and then we can record the signal digitally. So right now we're in the process of trying to leverage this technique to have this liposome fusion with exosome, and then to selectively enrich the breast originated EV, and to quantify and analyze markers on these cargoes. So we did actually go through a list of protein and nucleic acid biomarkers here. So the previous slide was on protein markers, and from this slide on is the microRNA markers. We did try to select our target from previous findings. So in our preliminary analysis, we used a subset of about 4 microRNAs just to testify this methodology. So these are the study we have generated using the non-TBI sample in green and the TBI sample in black. And we found an inverse actually relations between these panel of microRNA with the TBI injury. So the higher expression level in the non-TBI versus the lower level in the TBI group. So we have talked about EV, extracellular vesicle, as potentially a vehicle or cargo to carry diagnostic markers. I just want to expand from that a little bit because if we think reversely, not only we can retrieve EV and then study its content or surface marker, we can also use EV. We can actually derive EV from stem cells or even from neuron or glial cells, and then try to inject it into the body, and so that it can carry the pharmaceutical intervention we want to introduce. And because its property, its nature that it can cross the blood-brain barrier, this really makes it a very robust tool, as we said, not only to monitor what is happening in the CSF, but also trying to intervene in the brain. So I think the fact that I'm very excited about the extracellular vesicle research is that it just the free travel between in and out of brain and blood. I think the blood-brain barrier, it is a major barrier that have prevented us to utilizing or identifying biomarkers for brain dysfunction. But now with the technique advanced, that we can isolate and also bring these tests to near patient setting, I think we're getting closer and closer to have some markers and also possibly interventions that can be that we can do for the brain. So to summarize my talk, we reviewed current diagnostic approaches and their limitations, and we looked at emerging blood-based markers, including proteins, nucleic acid, and EV-associated markers. So we look forward to the emerging technologies that can enable the near-patient testing and also significantly improve our management for TBI. That concludes my presentation, and any of the patients or audience, if you do have questions, feel free to reach to me through this email address. Thank you very much for your attention.